Hello and welcome. My name is Martin Zach and I work as a philosopher of science focusing on immunology at the Institute of Philosophy of the Czech Academy of Sciences. And it is my great pleasure to introduce Professor Shimon Sakaguchi, who kindly accepted my invitation to visit Prague. And I'm grateful to the Department of Cell Biology of the Faculty of Science of the Charles University for providing the venue for today. Shimon is one of the world's most recognized immunologists who made the major discovery of regulatory T cells, which had been elusive for decades to the point that many prominent people in the field thought that they do not exist. And without these cells, modern immunology in general and immunological tolerance in particular are hardly conceivable. Some of you may wonder why a philosopher such as myself would invite an immunologist in the first place. In fact, it is a perfectly reasonable question, one which I'm going to briefly address in a minute. First, let me say a few things about Shimon. So Shimon received his MD and PhD from Kyoto University, after which he took on postdoc positions at Johns Hopkins University and Stanford University. He then landed the position of assistant professor at the Scripps Research Institute before returning to Japan in the early 1990s, to Tokyo, and later to Kyoto as a professor. In 2011, Shimon moved his lab to Osaka, where he is currently working as a distinguished professor at the Immunology Frontier Research Center at the Osaka University. Shimon is also the recipient of many prestigious international awards, as well as the Order of Culture awarded by the Emperor of Japan. While the comparison perhaps pales, it is my pleasure to announce that the Czech Immunological Society joins the appraisal of Shimon's achievements by awarding him the Garnet immunoglobulin. But before I invite my colleague Dominic Philipp to present the award, let me continue with a bit of an unorthodox introduction and address the connection to philosophy. No doubt, Shimon's work is the paradigmatic example of a brilliant experimental work. But the importance of his major contribution is not exhausted by it, nor should it be reduced to it. In fact, his insights are also deeply conceptual, reshaping our view of immunology. The experimental work complements the theoretical and conceptual work, which is no less important, as many leading biologists such as Sidney Brenner, Paul Nurse, Ruslan Medzitov, and many others repeatedly state. As Shimon puts it, what he is doing is tackling questions that have a philosophical flavor in the sense that they address deep conceptual problems. As a philosopher myself, I think that's right, and I firmly believe that Shimon is a great inspiration for philosophers of science who very closely engage with immunology, myself included, precisely because of the careful investigation of the conceptual landscape. And my colleagues in philosophy of science who do focus on immunology agree. In fact, Shimon has fruitfully interacted with a major philosopher of immunology before, and what is going on here is sort of a continuation of an existing small but growing international debate and collaboration between such philosophers and immunologists. Perhaps these words may come as a surprise to some. However, it's a sentiment shared more widely. For example, commenting on the 2017 Crawford Award, of which Shimon was the recipient, Claes Carré, who is the chairman of the Nobel Committee for Physiology or Medicine, said the following, and I quote, "The beauty of this prize is not only the fundamental nature of the discovery, it's also a very interesting and stimulating story from the point of view of history and philosophy of science," end of quote. Shimon's particular sense for philosophical thought also reflects the attitude of major immunologists of the past, the likes of Sir Frank Macfarlane Burnet, who received the Nobel Prize for Immunological Tolerance. After attending a symposium in Prague, Burnet wrote in 1965, perhaps hyperbolically, that he considered immunology to be more of a philosophical problem than a scientific one. A more modest claim, I believe, would be to say that philosophical insight is simply a part of science. It emphasizes the importance of historical awareness, attention to argumentative structure, careful analysis or proposal of concepts, frameworks, and theories, and has interdisciplinary nature. This is what makes philosophical thinking internal to immunology rather than something external, which is pretty much as it has been historically and still is nowadays. So finally, let me thank my colleagues who helped me to put everything together. First of all, Dominik Filip and Veronika Niederlova from the Institute of Molecular Genetics of the Czech Academy of Sciences, and Jan Dobeš, Jan Černý, and Marian Novotný from the Faculty of Science of the Charles University. And also many thanks to the Czech Immunological Society, represented today by Dominik Filip, who is now going to introduce the Garnet Immunoglobulin Award.
Dear Professor Sakaguchi, ladies, gentlemen, dear colleagues, dear friends, it is indeed my real pleasure to introduce Professor Sakaguchi. It's the first summer day today, and it's always connected to joy and happiness. And what is the better day to celebrate science and in presence of esteemed scientists such as Professor Sakaguchi? So I'm sure that those of you who are familiar with the work of Professor Sakaguchi would agree with me that to summarize his work in just a few sentences and his contribution to the world of science is practically impossible. We are lucky to have him here today because he's the discoverer of T regulatory cells, or Tregs. His work revolutionized the field of immunology. Tregs dampen immune responses and thus can act as brakes on the immune system, without which immune cells cause damage to self-tissues. Think of it as a car without brakes careening, wildly causing harm or even death to its driver or other cars on the roadway. Had Tregs not been discovered, we would likely know about them today because of omics technologies, but discoveries were made differently 30 or 40 years ago. Traditionally, by observing certain phenotype and designing experiments accordingly would provide hints and the nature of cells and mechanisms causing such a phenotype. While working as a postgraduate, Professor Sakaguchi became fascinated by a paper published by Nishizuka and Sakakura from Aichi Cancer Center in Nagoya that showed that removing the thymus from normal mice 3 days after birth resulted in autoimmunity. This indicated the existence of a type of immune suppressor cells. This was a turning point in his career which encouraged him to leave his current program and join the Aichi Cancer Center as a research student. Although the hunt for these suppressor cells was elusive, Professor Sakaguchi persevered and in 1995 demonstrated that a specific subset of CD4 positive T cells expressing the IL-2 receptor alpha chain CD25 could prevent autoimmunity. This was just the beginning of an exciting journey of Professor Sakaguchi into the world of Tregs. When IPEX syndrome, a monogenic disease that causes severe autoimmunity and allergy, was linked to FOXP3 gene, Professor Sakaguchi examined the relationship between FOXP3 and Tregs. His team was able to convert normal T cells into Tregs by expressing FOXP3 in normal T cells and found that FOXP3 is a master regulator of Tregs. This and many other findings altogether show that Tregs regulate not only autoimmunity, but also allergy, tumor immunity, microbial infection, organ transplantation, fetal maternal tolerance, immunometabolic diseases, and tissue repair. Because of the broadness and depth of these fundamental discoveries concerning Treg biology, it also provided a very stimulating story from the point of view of history and philosophy, as already mentioned by Martin Zach. In recognition of your discoveries and contribution to the field, I would like to present you Professor Sakaguchi on behalf of President Dr. Luca Vanucci and members of the Czech Society of Immunology, the Garnet Immunoglobulin Award, because we believe that you have pushed the boundaries of immunology beyond our wildest imagination. Congratulations and thank you for being with us today.
Well, thank you very much, Dominic and Martin. It's my great pleasure and honor to be here and also to be awarded the Garnet Immunoglobulin Award from the Czech Society for Immunologists. So it really encouraged me to further investigate how the immune system can be controlled in normal and the disease state, and then how we can treat, cure immunological diseases. So today, I'd like to talk about the immune tolerance and then regulatory T cells. Just a moment. Yes. So the definition of immune tolerance, that is immunological unresponsiveness to the antigen, that should be antigen specific, and a long-term unresponsiveness to the antigen to which the lymphocyte can potentially respond. So oh, yes. Well, so outline of my talk today is how important Treg is. They are immune tolerance and homeostasis. So let me talk about a brief history. Just already Dominic has kindly introduced to us. We have a history of Treg research. And then let me talk about this and also the molecular basis of their development and function. And second, these basic sciences, this is medical sciences, so we must also explain how the autoimmune diseases or other immunological diseases, how it happens in humans. And then in my case, how Treg contribute to the development of monogenic and polygenic autoimmune diseases. And third one is how to treat them, the immunological diseases by Treg. So let me start with kind of history. So as you see here, the immunological tolerance is not innately determined, but adaptively acquired. That is the most important concept in immune tolerance. So if the immune responsiveness, it is genetically determined, then organ transplantation is impossible. Always the host reject the other organs transplant. And then we can't cure autoimmune diseases unless we manipulate the genome. But what is important is it is adaptively required. And one of the important findings is made by the Czech immunologist Milan Hasek. Last year was his 70th anniversary of his discovery, and of 1953, of the chick embryo parabiosis. So on the same year, Peter Midaba, British immunologist, he showed that the mice, when newborn mice were injected with the lymphocyte from another strain of mice, and then they become tolerant because when you transplant the skin from the donor, and then they are not rejected. So it is the demonstration of the acquired tolerance. And then similarly, Milan Hasek, he showed with chicken embryo, just an parabiosis here, then circulation can be anastomosed. And then after birth, the chick didn't respond to immunization from each other. No antibody against the red blood cells. And later on, it was shown that the skin can be accepted from each other, like in mice. So conceptually, the first Macron Barnett, he showed that they proposed the concept that lymphocyte must have clones, and then they show clonal expansion. And then tolerance means that these clones are eliminated when they are exposed to the antigen in neonatal or fetal period. But then later on, in 1959, the Joshua Lederberg in the United States, he showed that not the age of the host, but an maturity of lymphocyte. So even adult animals, the immature bone marrow cells can become tolerized when it is exposed to the antigen. So this is the beginning of immune tolerance. So here, immune system, it can respond to non-self antigens such as viruses, bacteria, but it doesn't react with self. That is immunological self-tolerance. If we understand the mechanism of immune self-tolerance, then we may see how autoimmune disease happens, or how we can evoke good immune responses against quasi-self, so the tumor cells arising from self, or the ultimate goal of organ transplantation, that is, make the immune system look at non-self organ transplant as if it is self. That is the ultimate goal of organ transplantation. So allergy, environmental substances, they are not toxic, for example, pollen, but an immune responses against pollen causes allergy, pollen allergy. So against environmental substances, how we can induce tolerance. Or commensal bacteria in the intestine. We have more than 1 kilogram of commensal bacteria in the intestine, but we don't respond to them in normal state. But if the immune system responds to them, we have inflammatory bowel disease. So today, I like to use the autoimmune disease as readout of immune tolerance. And then how we can induce autoimmune disease in normal animals and how we can cure them by targeting regulatory T cells. So here, historically, the autoimmune diseases, how it can be the immunologically interpreted. So it is estimated that at least 5% of the population is afflicted with some sort of autoimmune diseases. So in the nervous system, multiple sclerosis, the thyroiditis, or the Graves' disease, thyrotoxicosis, or stomach with pernicious anemia, adrenal gland, and then insulin-dependent diabetes, type 1 diabetes in the pancreas. And then the SLE, systemic autoimmune diseases, and rheumatoid arthritis. So as I said, the first Barnett and then others, they postulated that the lymphocyte, when they recognize self-antigen at immature state, and then they'll be deleted. And nowadays we call them death by apoptosis. So this is true, really, in the bone marrow and also in the thymus. And then second, the mechanism is the inactivation. So lymphocyte can recognize self-antigen, but it doesn't become activated. It needs co-stimulation for activation, the antigen recognition plus co-stimulation, then they become activated. Just recognizing self-antigen, then they become inactivated. In immunology term, it is called anergy. And third mechanism is we harbor potentially hazardous self-reactive lymphocytes, but then their expansion or activation can be controlled by another subset of the T cells. Now we call them the regulatory T cells. So mechanism, that mechanism is suppression. The question was, which mechanism, if we manipulate, then can we produce these autoimmune diseases in normal animals? That was a kind of challenging question for immunologists for many years. So then the important feature of these autoimmune diseases, that is overlapping. So if you see the patient with type 1 diabetes, he or she may also have an autoimmune thyroiditis or autoimmune gastritis, stomach or adrenal gland Addison's disease. So this kind of overlapping is also common in animals. So NOD mice, it's a good model of type 1 diabetes. But when they become aged, say 1 year old, then you'll see some thyroiditis. The BB rats, it is also a good model of type 1 diabetes. But at the same time, they develop thyroiditis and gastritis. And a chicken, obese strain chicken, it spontaneously develops thyroiditis. But at the same time, they have the gastritis and other autoimmune diseases. So these findings together suggest that is autoimmune disease due to anomaly of the immune system or target self-antigen. So historically, how autoimmune disease happens, the people thought that maybe viral infection of the, say, pancreas, and then it may destroy the beta insulin-secreting beta cells, and then it may trigger autoimmunity. So the cause is in the target organ. But these findings suggest that, well, not the target antigen, but an immune system itself might have some anomalies. So second, so based on these findings in other animals, is a mechanism of immune self-tolerance evolutionarily conserved? That is second issue. And third one is, are the causes and the mechanisms are they common among various autoimmune diseases? So as I said, not the target antigen or target organs, but an immune self is the anomaly in the immune system. It may be responsible for various autoimmune diseases. So to address this question, there are two important experimental systems. One is the mouse system, the other one with rats. So normal mice. So just you should remove the thymus around day 3, and then some of them developed autoimmune gastritis. The ovulitis, this is the autoimmune disease in ovaries, and thyroiditis. This was first reported by Japanese two pathologists, Yasuaki Nishizuka and then Terio Sakakura in 1969. The second system is with rats, the normal rats. And then you remove the thymus around 6 weeks of age, and then every 2 weeks, 4 times, 200 Röntgen, 2 gray, X irradiation. And then some of them developed autoimmune thyroiditis and type 1 diabetes. So this was reported by a group in the UK, Penhale. So both systems, just manipulating the immune system, not target antigen. So thymus or perhaps X irradiation may affect lymphocyte. And it's enough for the autoimmune, the development of autoimmune disease. So what's the mechanism? So here, this is a neonatal thymectomy system. So bulb CA and C57 black 6 mice, all normal trained mice, but are genetically different. So bulb C mice, day 3 thymectomy caused gastritis, say 30%, oophoreitis, autoimmune disease in the ovaries, around 30%. But then day 0 thymectomy, within 24 hours after birth, almost nothing happened. So day 7 thymectomy, and also nothing happens. And if you change the strain to A strain, then you see 90% develop the oophoreitis, and then 10% gastritis and other autoimmune diseases. On the other hand, C57 black mice, they are very much resistant to the induction of autoimmunity by neonatal thymectomy. So day of thymectomy is critical, and also genetic background is important. So how this happens? So here, just removing the thymus, day 3 thymectomy can cause the autoimmunity, say autoimmune oophoreitis, around 90% in the A strain of mice. And then you inoculate normal T cells from adult mice before thymectomy, or 1 week after thymectomy, or 2 weeks, just 5 million T cells. Now you see the complete inhibition of disease development. So the mice become bigger, so 5 million T cells not enough 3 weeks later or 4 weeks later. So you'll see some development of autoimmune diseases. But this means that normal T cells, they contain a sort of immune suppressive T cells in normal state. That is the message from this experiment. So how we can interpret this? So normal thymus, it is producing T cells. It is produced in utero, and then day 0 mice are born, and day 3 if you remove the thymus, then some T cells cannot come to the periphery. And if you postulate that the T cells which have already migrated to the periphery, they may contain some dangerous autoimmune T cells. And then there's some the autoimmune preventive or suppressive T cells may come out from the thymus around day 3 on. Then, so if you inoculate normal T cells containing such cells, you can prevent autoimmunity. And then the question is, how we can differentiate between the autoimmune T cells versus regulatory T cells, immunosuppressive T cells, not by thymectomy, but directly if you remove this population from normal adult animals, whether we can induce autoimmune disease or not. So here the experiment we conducted in the past. So here normal mice, it has spleen. Spleen contain lots of T cells, especially CD4 T cells. And then if we try to remove the certain population by postulating that that population may contain immunosuppressive T cells. So by using various the cell surface markers. So first we use the CD5 marker. So we remove the CD5 high cells, and then transferred to nude mice. Nude mice is a congenitally T cell deficient mice. So B cells and antigen presenting cells, they are intact, but no T cells. So if you transfer by removing immunosuppressive T cells, and then these mice may spontaneously develop various autoimmune diseases. That was indeed the case. So about markers. So first we reported, and then a group in the Oxford, the UK, Fiona Powley and Dan Mason, they showed with nude rats, and then they removed the CD45RC low population, here, this population. And then they showed autoimmunity. And then the question was, why further removing the smaller population? And then if we can induce the more severe autoimmunity in a wider spectrum of organs, that is more specific marker. And then we came up with CD25, because CD25 is 10% of CD4 T cells, no CD25 positives in CD8, just CD4, and they are CD5 high, as shown here, and then CD45 RB low. And indeed, if you remove this population, just 10% of CD4 T cells, now you see various autoimmune diseases are shown here. So this is a stomach. And then this is an infiltration of inflammatory cells into the gastric mucosa. And then you can demonstrate the autoantibodies against gastric parietal cells. Parietal cells secrete hydrochloric acid and also the intrinsic factor required for absorption of vitamin B12. So autoimmune gastritis causes the pernicious anemia. Thyroid gland, and here thyroid follicles are destroyed, and then these mice develop the autoantibodies against thyroglobulin. This is pancreas, the islet. So the insulin-secreting beta cells, now they are under destruction. So you see lots of lymphocytes are coming in. And already other islets are destroyed. So actually this mouse developed type 1 diabetes. So just removing a small population of CD4 T cells, you can induce various autoimmune diseases, just similar to what we see in humans, and also inflammatory bowel disease. So how about why day 3 thymectomy? So if you look at the ontogeny regulatory T cells, so day 3, you see it is spleen. CD3 positive means the T cells. You see some T cells, but no CD25 positive cells. Day 7, some CD25 positive cells are now developed. And then day 60, they are about 10%, 5 to 10% of CD4, they are regulatory T cells. So in individual mice in the spleen, before day 3, you don't see them. But after day 3, very rapidly it increases in the periphery. So by thymectomy around day 3, you can shut off the migration of these regulatory T cells to the periphery. So neonatal day 3 in mice, now then this is a critical time. So just developing in the periphery, migrating from the thymus. How about other species? Rats, this is day 0. So to induce autoimmunity in rats, you must remove the thymus within 12 hours after birth. How about humans? This is 12 weeks in utero. So human, they are born in very mature state compared with mice. If you look at the mice newborns, the eyes they haven't yet opened, so it's very immature. But in humans, it's very mature. So, but this means that, as I'll come back to the Treg deficiency in humans later on, but then already in utero, autoimmune disease can start to develop. So in utero means without any environmental insult, no viruses or bacteria, just the intrinsic anomaly of the immune system, it's enough to trigger autoimmunity. So no environmental contribution. So just an anomaly of the immune system itself, it can cause autoimmunity. So here, the Treg develop, but then why day 3? So the T cells, the precursor come from the bone marrow, and then selected here. So those T cells highly reactive with the ubiquitous abundant antigen, they'll die by apoptosis. So clonal deletion is really it's working here. So McFarland-Barnett's clonal deletion, it is true. So 95% of the thymocytes in the thymus, they'll be deleted. But then the thymus really, but it really produces potentially hazardous self-reactive T cells. So why then the Treg? So thymus also produces regulatory T cells. And then one important feature of Treg, that is they become mature, functionally mature in the thymus. Then they migrate. So it may take time for them to become mature. So this is zigzag line, means it takes time. So that is the time difference even if both self-reactive T cells and Tregs are selected by by recognizing same antigen, a thymic antigen. But then their maturity, it takes time. So day 3 selectively reduce this population and trigger autoimmunity. So the self-reactive T cells differentiate into effector T cells, help B cells to secrete autoantibodies, and they help cytotoxic lymphocyte to kill the self cells, and then recruiting macrophages and others to cause the inflammatory inflammation. So this means that whatever it is, if the genetic anomalies or environmental insult, if they affect the balance between Treg and then effector T cells, self-reactive T cells, they may be causative of autoimmunity. So we've shown that already. I have told you that Tregs are sensitive to the ionizing radiation, X-ray radiations. And then say cyclosporine, this is chemical. So CD25 is the IL-2 receptor. I'll come back to this later on. And so cyclosporine, chemical. And then it selectively affects Treg and can cause autoimmunity. And certain viruses, for example, HIV virus, it infects CD4 T cells, especially Treg first. So the acquired immune deficiency, AIDS, and sometimes you see the autoimmunity because of the HIV virus, the selectively affect regulatory T cells. Now the question is, this scheme, it works in mice, in animals, but how about in humans? How we can demonstrate that this mechanism is important in humans? So there are many genetic anomalies in humans. And just a red color means the major one, which affect regulatory T cells. The most important one is FOXP3. So because there is a genetic disease called IPEX syndrome. IPEX means that immune dysregulation, polyendocrinopathy. So various autoimmune disease, endocrine autoimmune diseases spontaneously develop, including type 1 diabetes, thyroiditis, and so on. And then enteropathy, and inflammatory bowel disease against the commensal bacteria. And X-linked syndrome. So disease gene is on the X chromosome. So three features of this syndrome is autoimmunity. There are more than 80% of the patients develop type 1 diabetes within 3 years after birth, and more than 70% thyroiditis, and also other autoimmune diseases, including hemolytic anemia and then others. And then inflammatory bowel disease. So when the baby become feeded by milk of the mother, and then the intestinal bacteria start to develop, and then immediately they develop inflammatory bowel disease. And allergy, such as the dermatitis, or the babies when they take food orally, and then immediately they show the food allergy. So one gene can cause autoimmunity, allergy, and inflammatory bowel disease. How? And around the year 2000, the disease gene was positionally cloned by scientists in the United States. But the disease gene was designated FOXP3. It was found to be transcription factor. But how this anomaly can cause these immunological diseases? That was the question. So we postulated that perhaps the FOXP3 is specifically expressed by regulatory T cells. So scurvy mice, it has the same similar mutations. So if you stain FOXP3 intracellularly, so 10% of CD4 T cells express FOXP3. But the scurvy mice, no such cells. And then if you co-stain for FOXP3 and CD25, so interestingly, the majority of FOXP3 positive cells, they express CD25 in the spleen, also in the thymus. So previously we showed that by depleting CD25 positive cells, we could induce the various autoimmune diseases. This means that we remove a majority of FOXP3 positive cells. So we could induce autoimmunity. So now FOXP3 intracellular as an intracellular marker, CD25 as cell surface marker. These two markers together, we can define regulatory T cells, not only in mice, but also in humans. So just summarize this part of, let me summarize this part of my talk. So from the normal mice, if you remove regulatory T cells as CD25 positive cells, and then transfer the remaining cells into T cell deficient mice. So nude mice or skid mice, T cell, B cell deficient mice. These mice spontaneously develop autoimmune disease, IBD, and also allergy. And then in humans, the IPEX syndrome, so disease gene is on X chromosome. So usually patients are male kids. So HY, male is HY. So this is a sex-linked recessive disease. So the male kids develop severe autoimmunity, including type 1 diabetes, IBD, and severe allergy. But their mothers, so XX, so the half of the lymphocyte, because of Lyon's principle, X chromosome is the randomly inactivated. So half of the Treg intact, but then the other half abnormal. But then, so these intact ones really control effector cells. So mothers are healthy. So it means that in mice and also in humans, dominant self-tolerance is really working in the immune system. So here, another way of summarizing my talk. So the normal immune system, potentially hazardous self-reactive lymphocytes, they are present, but they are controlled not to be activated. So non-self antigens against viruses or bacteria, they are present. And if you remove Treg or reduce Treg, now the immune responses against self, now it appears. So you see the common autoimmune diseases such as diabetes and thyroiditis, they spontaneously develop. And also the immune responses against the commensal bacteria, it become enhanced. So you see immunopathology such as inflammatory bowel disease and also allergy to environmental substances. So this means that what is, well, perhaps philosophically, what is important is the border between self and non-self here, it is not fixed, but movable. So that is important concept. So as I told you that the immune tolerance can be acquired, but how it's acquired? So one interpretation is that this border, this can be, if it's put up here, then you can enhance immune suppression, so inducing graft transplantation tolerance. But if you put it down, then the immune response becomes enhanced. So not only autoimmunity, but also tumor immunity against quasi self-tumor antigen. That can be elicited, evoked. So that is indeed the case. So here, the same experimental system, T cell deficient mouse, and then they can be transferred with Treg depleted the spleen cells. And then if you inoculate the tumor cells, so here, tumor cells here, then tumor become bigger and then rejected. And after rejection, if you inoculate 10 times higher number of tumor cells, cancer cells, immediately they are rejected. So this means that they became immune to this particular tumor cells. So if you don't remove Treg, the tumor become bigger and bigger. And then the mice become, they will die by tumor progression. And if you put it, the 1-to-1 ratio, mix them. So the Treg become half in ratio from 4.4% from to 2.5%. But still, they suppress immune responses. So tumor become bigger and then all of the mice died by tumor progression. How about transplantation? Again, nude mice, T-cell deficient mice. So they don't have T-cells, so you can easily transplant and make them tolerant to the allograft. And then if you inoculate normal T-cells, so 100% the accepted skin, they become rejected. Within a month, all the graft can be rejected just by transferring naive T-cells alone. But if you first transfer regulatory T-cells, so they come to the graft site and then proliferate. And then 1 week later, if you inoculate normal T-cells, then the graft rejection can be retarded, prolonged. And then 20% become permanently tolerant. And if you increase Treg to say 1-to-3 ratio, now the 60% become permanently tolerant. So the graft tolerance can be induced. So just without any immunosuppressive drug, but just expanding naturally present regulatory T cells, you can induce graft tolerance. And then what is important here is that, so these mice, so they are tolerant, the Tregs are there, and they're suppressing the effector T cells. And then if you, again, retransplant the same graft from the same strains of mice, or a third-party irrelevant graft, what happens? So original square one, this is the original graft, it's stable, stably tolerant. But the same, genetically same graft, new one, they become rejected and they become stabilized. Now they become permanently tolerant. The third party one, immediately rejected. So this means that those T cells which can reject graft, they are present in this tolerant mice. Just they are suppressed not to become activated or expand. So this is really the dominant tolerance. Like self-tolerance, the self-reactive T cells can be suppressed by Treg. And graft tolerance, graft rejecting T cells, they are suppressed by Treg. The same mechanism. So in humans, so this is one example. So this is, living donor transplantation, liver transplantation. So this boy, when he was born, he showed the congenital bile duct atresia. So the part of the mother's liver was transplanted to this boy, and then the heavily treated with immunosuppressive drugs. And unfortunately, he showed very severe infection because of the immune suppression. So the physicians, they stopped medication. But he didn't reject the mother's liver. And 10 years later, any immunosuppressive drug, he is playing baseball. So we have many such patients. Many of them are transplanted liver when they were babies. So here, the tolerant group, and if you look at the biopsy, so this is a transplanted liver, and this is portary area. You see CD4 T cell infiltration, CD8 T cells, and also regulatory T cells, FOXP3 positive cells. And if you enumerate the number of FOXP3 positives, calculate, and then these tolerant people, the higher number of regulatory T cells in the tissue, and their function. So tolerant Treg, and then responder T cells from a healthy donor. So this is alloantigen. So they responder T cells, they respond and then proliferate. But the regulatory T cells, when they came from the donor, tolerant donor, and then these Treg recognize this donor antigen and suppress immune response. So this white column means that they can suppress the antigen-specific fashion, the donor-specific immune responses, but not the third party. So these findings together Now we know that the normal thymus, it produces regulatory T cells, and then it controls the various immune responses. So this anomaly, reduction in number or function, can cause autoimmune disease, allergy, and immunopathology such as inflammatory bowel disease. And then by reducing them, you can strengthen cancer immunity, or by expanding them, you can induce organ transplantation tolerance. And then also fetal-maternal tolerance. Why the mothers they don't reject the fetuses in pregnancy? This is still a big question in immunological sciences. But one interpretation is that the Treg can control the rejection or abortion of fetuses. And then we know that immunometabolic diseases, for example, obesity or atherosclerosis in the vessel, they have an inflammation, and then the Treg can come to the fat tissue and suppress the inflammation. And then this is important for prevention of type 2 diabetes or obesity and atherosclerosis, and also tissue repair by producing various cytokines, including the tissue repair substances. They can contribute to tissue repair. Now, let me move on to the molecular and the cellular basis of this immune tolerance. So as a phenomenon, the Treg can control or induce tolerance, but how? That is the question. So let me talk about several molecules, such as IL-2, CD25. This is IL-2 receptor, CTLA-4, and PD-1, PD-L1. So here, FOXP3, the most important function of FOXP3 is if it is expressed in normal T cells, conventional T cells, it can convert normal T cells into Treg, possessing Treg-like suppressive activity. So by retroviral vector, you can express FOXP3 in conventional T cells. And if you co-transfer with the Treg depleted spleen cells, so without such cells, it can cause colitis, inflammatory bowel disease, or autoimmune gastritis. But if you co-transfer with the FOXP3-induced conventional T cells, now you can prevent disease development. Just an empty vector one, no suppression. So here, normal colon, colitis can be prevented by FOXP3 expressing the conventional T cells, and then empty vector, no such suppression. Normal gastric mucosa, gastritis can be prevented. So FOXP3 can confer suppressive activity on the normal T cells. How? So FOXP3 is a transcription factor. So it controls the hundreds of genes, either activate or depress. So which gene downstream of FOXP3 is important? The simple question is, the downstream gene, which one, if it is deleted or overexpressed, and then does it produce FOXP3-like suppressive activity, FOXP3-dependent suppressive activity? So like FOXP3 deficiency causes autoimmunity, and then which one downstream, which gene can cause similar autoimmunity? Not many. One group is IL-2. IL-2 itself, or IL-2 receptor. CD25 is the IL-2 receptor alpha chain, CD122. This is IL-2 receptor beta chain. And then another one is CTLA-4. So let me talk about an IL-2 and IL-2 receptor, and then CTLA-4. So FOXP3 depress IL-2. So one feature of Treg, that is, after activation, even after activation, they don't produce IL-2. And then another feature of Treg is the IL-2 receptor composed of alpha, beta, gamma chain, which is a high affinity IL-2 receptor. the high affinity IL-2 receptor is constitutively expressed by Treg. So the normal T cell, conventional T cells, only when they are activated by antigen, then they express the high affinity IL-2 receptor. But the regulatory T cells, they are already expressing the high affinity IL-2 receptor in the thymus when they are produced. So here, CD25 is the IL-2 alpha chain positive cells. They are also expressing IL-2 receptor beta chain, and also the gamma chain, alpha, beta, gamma. So already in the thymus, they are expressing the high affinity IL-2 receptor. And so they don't produce IL-2. So without IL-2, they cannot survive. So just a neutralizing IL-2 in the normal animals, just one injection of anti-neutralizing antibody, then you see the regulatory T cells, they'll die, as shown here. So this means the IL-2 neutralization can cause autoimmunity. So just twice, if you inject IL-2 neutralizing antibody, and then 3 months later, you see autoimmunity. This is the autoimmune gastritis, and then this is a titer for the gastric parietal cells. And then if you inject into NOD mice which are prone to develop type 1 diabetes. So type 1 diabetes become more severe, thyroiditis become more severe, and the lacrimal gland or salivary gland adenitis, like a human Sjögren's syndrome, you see this kind of inflammation. So IL-2, well, this is the important cytokine in immunology. This is only cytokine. When you neutralizing it, it can cause autoimmunity. How about the CTLA-4? So FOXP3 positive cells, they are expressing CTLA-4, highly expressing CTLA-4. And then if you make a conditional knockout mice, the only Treg, they don't express CTLA-4. CTLA-4 conditional knockout mice. And then these mice spontaneously develop very severe autoimmunity. Like it is myocarditis. So the infiltration of a lymphocyte, inflammatory cells into cardiac, into the heart, and then cardiac myocytes are under destruction. And then, so the heart become bigger, and then, so this is the infiltration of the inflammatory cells. And then not only myocarditis, but also other autoimmune diseases spontaneously develop. If you look at the serum IgE, several hundredfold higher. So they are prone to develop allergy. And then CTLA-4, as you may know, anti-CTLA-4 antibody, ipilimumab, it is under use for the human cancer immunotherapy. So if you inoculate the tumor cells, and the red one, these mice reject the tumors, but the control mice, they'll die by tumor progression. So how about the CTLA-4 antibody? Like an IL-2 neutralization, just twice, if you inoculate anti-CTLA-4 antibody, and 3 months later, and you'll see the severe autoimmunity. This is autoimmune gastritis with high titer of anti-parietal cell autoantibodies. And so CTLA-4 is important for regulatory T cell function. How? So here, red one is the CTLA-4 knockout Treg. They don't show in vitro suppression. So blue one is normal Treg. So responder T cells, they proliferate. And if you co-culture with the normal Treg, so 1:3, 1:1 ratio, the responder T cell proliferation, it can be suppressed. But on red one, CTLA-4 knockout Treg, they don't suppress the immune response. How? So here, the middle one is antigen-presenting cells. So CTLA-4 is red, and then CD80, 86 green. So what happens is that when Tregs are activated, they make cell contact. And then what happens is that CD80, 86 become aggregated, and CTLA-4 become aggregated. And then these the CD80, 86 become transferred to Treg and then endostased. So on the cell surface, CD80 and 86 become reduced in expression. So by the FACS staining, so here the activated dendritic cells, and then they are expressing the high CD80 high and the total PDL1 high. And then free PDL1, not many. And if you co-culture with Treg, so CD80 expression become lower, as shown here, because Tregs are Treg deprive, they snatch out the CD80, 86. So the expression level become lower. And importantly, free PDL1, it can interact with PD1. Their expression become upregulated. So here, what is known now is that the CD80 and the PDL1, so they are associated in normal state. And then if you Treg remove CD80, now the PDL1 become free. So these free PDL1, not associated one, free PDL1 can interact with PD1 expressing cells and control them. So this means that when Treg downregulate CD80, 86, it can suppress the activation of naive T cells. And also, by increasing free PDL1, now it can, the Treg can control the effector T cells expressing PD1. So this is a summary of this part of my talk. The FOXP3, it replaces IL-2, but upregulate IL-2 receptor. So this means that the Treg can absorb IL-2 from the surroundings or deprive IL-2 from responder T cells. So this is one mechanism of suppression. And also the FOXP3 upregulates CTLA4, and the CTLA4 interact with CD8086 and downregulate CD8086. And then by this mechanism, free PDL1 become upregulated. So for responder T cells, they are deprived of IL-2, deprived of co-stimulation. And then also the receive the PD1 mediated the negative signal. So this is a mechanism of suppression. And then Treg become further differentiate. They also produce IL-10 or TGF-beta or other immunosuppressive cytokines. So then the next question is, what happened then? These responder T cells. So Treg suppress APC. And then what happened to these responder T cells? Just they are suppressed, or do they become, their cell fate become changed? So what happened to self-reactive T cells subjected to Treg-mediated suppression? So here, this is human peripheral blood, and then we are using melan A antigen. So melan A is a self-antigen, and then expressed by melanocyte. This is melanocyte differentiation antigen. And this is also target antigen of vitiligo vulgaris. This is the skin autoimmune disease. And then it is also a tumor-associated antigen in melanoma, malignant melanoma. So here, normal healthy blood lymphocytes, and then removing Treg, and then stimulate with melan A peptide. And 1 week later, you can detect melan A specific lymphocyte by tetramer staining. So as you may know, this is TCR. TCR can be detected by MSC plus melan A peptide. This is tetramer. And then so you can detect antigen-specific T cells. So here, without Treg, now you see 2.7% expansion of melan A specific CD8 T cells. And if you add regulatory T cells, now expansion can be suppressed, so in a dose-dependent fashion. So these melan A reactive T cells, what happens? So by removing the Treg, melan A specific one can expand. By putting back Treg, it can suppress the immune response. And the fact, and the issue, these two populations proliferating melange specific T cells versus non-proliferative one here, A and B. What's the difference? So what the difference is, if you de-stimulate them, secondary stimulation, so these guys can proliferate, CFEC diluted, but peak B, they don't proliferate. And by stimulation, they don't produce cytokines, including IL-2. So this is the definition of energy. So they don't proliferate, and then they don't produce IL-2 or other cytokines. So in the presence of Treg, their proliferation suppressed, and then also they become anergic. And then one thing what is important is, you can also compare the TCR affinity by tetramer staining. So these tetramer high cells, they are high affinity. And then tetramer staining low population here, they are low affinity. So you can compare A and B, and then what you see that A show higher affinity than B. So TCR molecule itself or CD no difference, but the affinity is different. So they are high affinity ones, and then these guys are low affinity ones. So and then in vivo, in humans, so here healthy donor, you can detect melange specific T cells. They are present in our blood. And then in vitiligo vulgaris, and then in autoimmune disease, they have expanded. And then in vivo, if they compare the affinity, and then healthy donors, they show lower affinity, human donor tetramer positive cells. And if you stimulate human donor tetramer positive cells, they don't produce IL-2. So in vivo situation, self-reactive T cells are present there, but a majority of them are anergic. So these findings together, now we know that Treg controlled APCs, and then conventional T cells under Treg mediated control, CD80-86 become low. And then if the T cell affinity of responder T cells, if it is high, then they may die by apoptosis. If it's an intermediate affinity, they become anergic. If it's a low affinity, they are not dangerous, so they stay naive. And if you remove Treg, the APC can be activated, CD86 high, and then conventional T cells become activated whether they are low or high T cell affinity and causing autoimmunity. So this means that suppression, energy, and apoptosis or cell death. So I have told you that there is three mechanisms have been the kind of main interpretation of immune tolerance. Now what we know that they are linked in immune tolerance and suppression. So now let me move on to the next topic, that is human autoimmunity. So as I told you, the 5% of the population is afflicted with autoimmune disease. And then IPEX syndrome, the FOXP3 mutation can cause really the autoimmune diseases, type 1 diabetes. But how about the common autoimmune diseases, the popular ones? The Treg important or not, or different mechanism? So the question is, we know that by single nucleotide polymorphism, GWAS, and then lots of autoimmune SNPs now we know that they contribute to autoimmunity. And the question is, these autoimmune SNPs, are they really, do they really affect Treg development or function? So here, the important clue is a super enhancer. So this is in the genome. And then here, this is the enhancer region and control the gene expression. But in the genome, there are many enhancers called super enhancers, where a dense accumulation of transcription factors, and then a histone modification for gene activation, or P300 binding, and so on and so forth. And then these super enhancer sites strongly control the gene expression. And then as a result, they determine cell fate or cell identity. So this means that Treg-specific super enhancers, if they are present, they strongly control Treg-specific gene expression and also the epigenome. So here, this is Treg and the conventional T cells. So CD28 and CTLA4. So conventional T cells. So this is H3K27 acetylation. So it is a marker for active enhancer. No difference. But here, CTLA4 locus, the conventional T is not activated, but the Treg highly activated. So this is called the Treg specific super enhancer. And we found that the 66 and Treg specific super enhancers in the genome. For example, this is a spine enhancer lesion. It includes CTLA4, yes, FOXP3, and then IL-2 receptor alpha chain, CD25. So many Treg signature genes, they do have the Treg specific super enhancers. And then the question is, autoimmune SNPs, are they located at the Treg specific super enhancers? That was indeed the case. So we found in humans 21 Treg specific super enhancers, such as FOXP3, CTLA4, CD25, and then many autoimmune SNPs, they accumulate at the Treg specific super enhancers, such as the vitiligo, Graves' disease, multiple sclerosis, type 1 diabetes. So many autoimmune diseases, as you see in IPEX syndrome, in normal, common people, they have an autoimmune SNPs which has propensity to affect the Treg function and development. So this is the first report of the GBAS study published in 2007, 1,000 of the control and then the disease samples. And then this is type 1 diabetes, and then 2 genes, CTLA4 and IL-2 receptor alpha chain, CD25. They have autoimmune SNPs, but then at that time, we didn't know whether these polymorphisms affect effector T cells, CD25 positive, CTLA4 positive, or regulatory T cells, which are also CD25 and CTLA4 positive. Now we know that these polymorphism variants really predominantly affect regulatory T cells. So here, just similar to IPEX syndrome, there are now the category of disease called T-regopathies. The mutations of Treg signature genes, such as FOXP3, CD25, and CTLA4, causes monogenic autoimmune diseases by affecting Treg development and function. And also common autoimmune diseases, genetic polymorphism, in particular in Treg-specific CpG hypomethylated lesions. They, for example, FOXP3, CD25, CTLA4, affect the development of naive Treg cells, and then make the host susceptible to various autoimmune diseases, such as type 1 diabetes and rheumatoid arthritis. So then the question is, the common autoimmune diseases, how environmental factors affect the autoimmune disease. So one important report by eminent French immunologist, Jean-François Bach, unfortunately he passed away last year. So he published in New England Journal of Medicine that in developed countries, within these 50 years, the infection, the rate has become lower. So including rheumatoid fever, measles, mumps, all become lower. But then at the same time, the autoimmune disease and the allergy, like asthma, and also inflammatory bowel disease, such as Crohn's disease, are significantly increasing. So this is the hygiene hypothesis. So the society become clean. Now certain autoimmune diseases, they increase in incidence. And then exactly these diseases are caused by Treg anomaly, like IPEX syndrome, autoimmune disease, asthma, allergy, and inflammatory bowel disease. So the increased society, so the no significant antimicrobial immune responses. So you don't have to control immune responses too strong or too weak. And then in that case, Tregs are not well trained and not so strongly control, efficiently control immune responses. So that may contribute to disease development. So here, to summarize this part of my talk, so Treg anomalies, if it becomes a bit weaker, then the suppression becomes weaker, then those genetically susceptible people, they develop autoimmunity. So individual, they have a different repertoire of the immune responses. For example, this individual, if you remove Treg, he develop or she develop type 1 diabetes. The second one, develop a thyroiditis gastritis. But the third one, no autoimmunity. Like in the beginning, I talked about a neonatal thymectomy system. Just like in humans, the genetic background and the degree of Treg anomaly, and then they make the individual susceptible to autoimmunity. And then just innate immunity, stimulation of innate immunity, the usual infection. And then just one push from the back, and then you see the autoimmune disease become overt. So that is our understanding of how autoimmune disease develop in humans. And then just briefly, let me talk about then how to treat them. So most important one is antigen-specific immune suppression. So steroid or tacrolimus or cyclosporin A, all are the antigen non-specific suppression. So it makes the patient become susceptible to infection. So in modern immunology, what is required is how to induce antigen-specific tolerance or immune suppression. So here, this is current ongoing trials. So how to increase Treg? So as I told you, so Treg are constitutively expressing the high affinity IL-2 receptor. So this means that low dose IL-2 is enough to expand them. So effector cells, they need high IL-2 concentration, but just low dose. Then you can selectively expand a Treg and then make a Treg dominant state. And the second one is the Treg, they are circulating in the blood, so you can take them out, the ex vivo, and then stimulate them, expand them, and then transfer back to the patient. And this is adaptive cell therapy. Or even you can put in a CAR construct into Treg, and then they make them migrate to particular inflammation site. That is, they are under study or under clinical trials now. But they are not antigen-specific suppression. So ideally, what we think is what is important is effector T cells, they causing inflammation or causing autoimmunity. So they have antigen specificity. If they can be converted to Treg with such antigen specificity, that is the ideal way of the antigen-specific suppression. So this is in vivo, and then you can take effector T cell out and then convert them to Treg, and then transfer back to the patients. So we are now working on this side of the antigens, how to produce antigen-specific Treg from effector T cells. So this is our trial. We screened more than 5,000 compounds for the FOXP3 induction. So this is a synthetic substance. So here, the naive T cells and the effector T cell and the memory T cells, they are mediating disease. So naive T cells, you can put in the TGF-beta, you can induce FOXP3. And without TGF-beta, this substance called AS2863619, abbreviated as the AS compound, it can induce FOXP3. And also, in effector memory T cells, you can induce FOXP3 positive cells, but TGF-beta cannot. And then CD8 T cells, you can induce a Treg here by this compound. So this compound, this is a CDK819 inhibitor. So TCR signal activates CDK819. This is a regulatory module of mediator complex which controls the transcription of various genes. So CDK819 inhibits STAT5. So in activated T cells, so it inhibits FOXP3 expression. But here, if you make a CD819 inhibitor, then STAT5 become activated and the FOXP3 gene is on. So they express their FOXP3. So here, so this substance, and then together with antigen stimulation. So in this case, the T cells are all specific for over-albumin specific transgenic T cells. And then stimulated with OVA peptide, and then AS compound in the presence or absence of AS compound. And so without AS, nothing happened. But the AS compound, and then together with antigen, and then antigen, the TCR positive cells, they become FOXP3 positive. So only antigen stimulated T cells become Treg. In vivo, so this is all T cells are specific for over-albumin. And then here, OVA immunization plus AS compound orally taken. And then these OVA specific T cells become Treg, as shown here. And then these induced Treg, they are just similar to the natural Treg, and they show the epigenetic changes. This is the FOXP3 gene locus Treg specific demethylation in natural Treg and also AS induced the Treg. So by oral treatment with AS compound, you can suppress the contact hypersensitivity. And then if they are treated with AS, now the ear swelling can be suppressed. And if you remove Treg from that condition, now the inflammation cannot be suppressed. This is type 1 diabetes. So just orally administer this compound, then suppress the type 1 diabetes. So this is histological score. So AS compound, almost all islets are intact, no inflammation. So these findings all together, now what we are working on is for antigen-specific immune suppression, the effector memory T cells mediating autoimmune disease or graft rejection or allergy, they can be converted to antigen-specific regulatory T cells. So they can be used in vivo, just a treatment, oral treatment, or in vitro, and make them, and then use them for adoptive cell therapy. So this is the last slide of my talk. What I have talked about today is the normal immune system. It is equipped with the T cells, the specialized for immune suppression, called regulatory T cells. They are expressing FOXP3, and then also they can be induced from normal T cells, for example, in the intestine. But also we can convert effector T cells into Treg. And then just I've told you that the border between self and non-self, it can be movable. So by the up means strengthening Treg, you can treat autoimmune disease allergy or IBD or transplantation or fetal maternal tolerance and so on and so forth. And also by putting down the border or the reducing Treg number or weakening Treg suppressive function, you can enhance the immune responses like cancer immunity or chronic microbial infection. So that is by targeting Treg, either way, immune suppression or immune enhancement. In the clinical setting, we are now trying to both ways to treat immunological diseases and also treat cancer. So the many students and postdoc, and they have been involved in these studies, which I have talked about today, and the current colleagues and collaborators, for example, AS compound or the other project. Thank you very much for your attention.
So yeah, this one also works. So we have room for questions. I would like to see some hands up if there are questions. Yes.
Thank you for your very clear talk. So I would like to ask regarding the Treg generation in the thymus in the perinatal life, basically. So do you think that there are different waves of like Treg generation in the thymus that give rise maybe to functionally a little bit different Tregs on like different days after the birth?
Well, it is still controversial. How developing, let's see. So developing T cells, they not conventional T cells, but going to Treg, whether this is really affinity for thymic self-antigen expressed or something else. The current dogma in this field is that the intermediate affinity T cell receptor expressing cells, they somehow differentiate into Treg by expressing FOXP3, and then low one become naive. The high affinity one, they may die by apoptosis. That is a kind of a dogma, easy to say, but the actual molecular events, still we don't know. We have different ideas, but it's a kind of a heretic one. So I should say it is important theme of research, yes, but I should say it is still controversial, not much known.
Thank you.
Hi, thank you for the beautiful talk. I have a very general question for you. I see that you showed very nicely that these mistakes in T regulatory cells, they lead to autoimmune diseases, many of them, but they are mostly organ specific, you know, such as diabetes and some other, and it is not the case with systemic autoimmunity. I mean, lupus and rheumatoid diseases, they are not so directly connected. What do you think? I think it's the difference.
Mm-hmm. Yes, that's an important question. So let me. So I have mainly talked about the organ-specific ones, but if you remove Treg, in our first report in 1995, we found also systemic autoimmune diseases like SLE, and some of them developed arthritis, so joint becomes swollen. So still, I believe that there are common bases, the SLE or systemic autoimmunity, and then organ-specific ones. Actually, if you look at the various animal species, for example, in dogs, the dogs frequently develop an SLE plus the thyroiditis. So there may be some differences in phenotype, but Treg anomaly or anomaly of immune regulation contribute to both organ-specific and then systemic autoimmunity. So this is also supported by GBAS studies. So the the SNPs for rheumatoid arthritis, for example, it also affects Treg. Then recent reports.
Thank you very much for a very nice talk. It was brilliant. So I would like to ask you, so why some autoimmune disease develop in elder stage in life and some in childhood? It's like based on the more inflammatory state of elder people that in childhood, in Treg.
Well, so perhaps the question is, some autoimmune diseases are more frequent than others. And then, yes, that's true. So that is, one is the MSC or HLA haplotype. So MSC, what kind of a cell peptide they can present. So MSC haplotype can determine the phenotype of autoimmune disease. And then also, but basically, like an IPEX syndrome, severe Treg deficiency, and within 3 years after birth, if you are Caucasian, then more than 80% develop type 1 diabetes. So meaning that the Caucasian, well, we are Asians, they are a bit lower, but all of them, you are susceptible to type 1 diabetes. This means. So type 1 diabetes, it's very easy to detect. But other autoimmune diseases, it may take time to become manifested. So that is another difference. And so I believe that almost every tissue, for example, we've shown that the heart, myocarditis, autoimmune myocarditis, and we also induced even the bladder. You can induce autoimmune cystitis or whatever by manipulating Treg. So almost every tissue, every organ, by targeting Treg, we can induce autoimmunity. Of course, we must a bit changing the genetic background, and then you'll see various autoimmune diseases. So that is the basic, our basic understanding.
Thank you very much for a very nice talk. I got 3 questions. First is if you could comment the history, because I am old enough to remember time of the era or period of suppressor T cells in 70s, and then it was refused, and then new era. So this is first. Second is connected. Why do you think that in germ-free conditions, these models of human diseases, autoimmune diseases, with exception of not mice, diabetes, are not developing autoimmune diseases? And third one is if you could explain or comment the relationship between central tolerance and peripheral occurring on mucosal surfaces.
All right, okay. Well, to the first question about suppressor T cells. So here, a young audience, you don't know about suppressor T cells. But in the 1970s, there was the popular topic of immunology research was suppressor T cells. So suppressor T cells at that time, they are CD8. And then if you immunize the mice certain ways, you can induce suppressive T cells. And then, but that research, the collapsed pen, the molecular biology, they scrutinize the suppressor T cell dependent genes and so on. They no support from molecular biology. So what's the difference between old suppressor T cells and the current regulatory T cells? That is the question I'm asked now. So we think that our success is we worked on autoimmune disease. So whatever the mechanism is, by manipulating the immune system, disease happened. So this is always, we can come back to this finding. But on all days, CD8 suppressor T cells, just if you immunize, and then they measure the titer about antibody or something, it's not reliable, I should say. So the disease, this is really not the immune response. This disease means immune response really destroy tissue and cause disease. So, and then I'm originally a pathologist. So always we come back to that finding, whatever the mechanism is. So, and then we came up with regulatory T cells. So perhaps that is a big difference of all the suppressor T cells. And then all the suppressor T cells, they fail to show their importance in real clinical setting. So if all the suppressor T cells can explain certain autoimmune disease, or if really can enhance tumor immunity, or induce the transplantation tolerance, perhaps their science can continue. But actually, all they failed. But we had a kind of success in this. And also, to us, the yes, and germ-free condition. Well, so what I've shown today is that without the environmental insult, well, germ-free condition, and then still we can induce autoimmunity. So that is what we see. But this also means that the microbes can really affect the immune system and modify the disease progression or incidence. That is also true. So it's currently it is a kind of a hot topic in immunology, how commensal bacteria or microbes can contribute to immune responses, for example, anti-cancer immunity or others. So we haven't touched much yet on that issue, but I do believe that the microbial environment, it can affect, influence the immune response and tolerance, yes.
Thank you very much for a wonderful talk. It's an honor to have you here. So my question is a bit regarding the, let's say, innate and peripheral or acquired regulatory T cells. What do you think, what is your opinion? Because there's been some debate about this. What is your opinion of the role of the transcription factor Helios and its ability, or its there has been various discussions about if it is possible then to distinguish the peripherally generated Tregs from the thymic Tregs using it or not. And I would be very interested in your opinion on this.
I see. Well, so Helios is another transcription factor which has been shown to use for define a natural Treg, thymus-derived Treg versus peripherally produced Treg. So still it is useful. And then, but the real function of Helios, so as you said, it is still controversial. And then some people claim that Helios is important for stability and of gene expression. but then also Helios knockout, it is a bit controversial. So, well, as a custom, we are always looking at Helios, but I'm very much hesitant to say that it is a good marker for a natural Treg or something else. Yeah, I don't have a good answer to your question, specific answer to your question. I'm very sorry about that.
Thank you. It's basically what we in our group were thinking as well, but I was very interested in if you have a better answer.
Well,
so, thank you. Thank you very much.
And also, you asked me about the central and peripheral tolerance, and how it's affected. And then the central tolerance, it is really important. It happens really abundant, ubiquitous self-antigens. It really happens. The ubiquitous means that the enzymes for respiratory chain or glycolysis or these or abundant blood proteins, say albumin, all happens. If they circulate to the thymus, you can induce the central tolerance. But usual self-antigen targeted in autoimmune disease, for example, insulin or others, they are present in the periphery. And also they develop when they developmentally appear. And this tiny amount of self-antigen in the periphery, do they circulate to the thymus and then cause central tolerance? Well, perhaps very high affinity T cell receptor expressing cells, they can be deleted. But the majority of them, they are not deleted. They are present in the periphery. So now it is because of technology advancement, you can easily detect insulin reactive T cells in the normal healthy individuals. If you enumerate the 1 per 1 million lymphocyte, that frequency, they are reactive with, say, insulin or keratin or other authentic self-antigens. So they are present in the periphery.
Hello, thank you for the great talk. I got intrigued by the involvement of Tregs in the hygiene hypothesis, and I think it's really great. But I was wondering, what do you think, how does it mechanistically work that Tregs need to be probably trained, and we don't have them trained sufficiently in the developed world nowadays? Is it true, for example, I don't know, that we need a lot of IL-2 from the activated T cells during infection and we don't have enough, or excess TGF-beta, or maybe also the microbiome, as you mentioned now? What do you think?
Well, that's an important question. So still we don't know the mechanism, molecular basis of the mechanism. But generally, I believe that is true. For example, in my lab, I have many students, and then now 80% of them, they have a pollen allergy. But when I was born, the kids around me, nobody has an allergy. Nowadays, allergy is very frequent. And so what's the difference? So one reason is the society become the clean, and then so the Treg. Well, let me see. So if you go to the Africa, still children, they may die by infection, especially diarrhea or alimentary tract infections, and then the Stone Age environment. So I do believe our immune system is adapted to the Stone Age environment and not the developed the modern society. So that is the one thing, the kind of a contradiction of the between the environment and then our immune system. So as you suggested, we need a more precise investigation. What the difference in the hygienic society. Just simply, we can say that Treg are not much trained. Also, the effector cells not much trained. And Treg, they don't have to suppress immune response because the immune response is not so strong. So that kind of balance is very weak, very fragile. But if you live in, say, Africa or somewhere, always you are infected. And then if the immune response should be kind of not too much, the Treg also must be strengthened. And a strong balance between effector cells and Treg, that is more stable compared with the weak immune response versus weak suppression. And then that balance is kind of precarious, not so stable. So that is all I can say at this point. But I agree with you that there must be, we must know some cellular and molecular basis of this. Epidemiologically, yes.
Yeah, I see a question over there, but we are also closing, approaching our time. So,
thank you very much. I have a following question. So do you think like the dysfunction of the Treg is more like quantitative, that there is like smaller frequencies of those Tregs because those are usually really limited by the IL-2 or another like survival factors, or is it more like qualitative, or like the functional of the Tregs is impaired with the, I don't know, CTLA4 or production of the cytokines? So do you think it's more like we have like different numbers of those Tregs with different repertoires, or we have like similar numbers of them but their function is not just as good as those trained from like some infections?
So your question is for particular situation, Treg number or size?
Yeah, because as what we can see here on this slide, it's like the decreased thresholds of like the Tregs function. It's really nice. It's because there is like smaller amount of Tregs that have the smaller effect, or it is like the number is the same, but just those Tregs don't have the effect. It's needed. What do you think?
Well, I think it's both. So number-wise, it is true that I measure the Treg number in my lab. in my lab. So I'm the lowest number, perhaps susceptible to autoimmunity, perhaps. And so the number becomes a bit variable, but usually around 10%. And if you become aged, it a bit increase, say from 10% to 15%. And then their repertoire may be different depending on your experience. For example, I looked at the one person and then 1.5 year period, and then their Treg repertoire, whether it will change or not. And it really changes. So we found certain clones in the naive Treg fraction. And 1.5 year later, that population, that clone now moved to effector Treg population and expanded. So this means that not only a total number, but also a repertoire. That is important for, this is kind of a bit simplified, but you are right. Not only number, but also the repertoire, which dominant specificity are there. That is also important. And I believe that the autoimmune disease patients, the self-antigen specific Treg, they have expanded, but not enough to control disease. So what we are doing is how we can then expand more or strengthen more to treat disease.
Thank you very much.